Hi everyone. This is the second mini lecture in Chapter 9. We're covering physical and chemical controls of microbes. In this mini lecture, I'm going to go over a little bit of the physical controls that are used. There are 6 main controls that we use for physical control, the first of which is heat. And at elevated temperatures, very elevated, we have microbicidal, which will kill the microbes. And at lower temperatures, we have microbistatic, which, if you remember, static means keep them from really growing or growing more, but it is not going to kill all of them. There are 2 different ways that we can use this physical control of heat. The first is moist heat. So that would have steam or hot water, boiling water involved. And this is much more efficient than dry heat. And then, so we have dry heat as the other one, which is hot air or an open flame. And these actually are higher temperatures that we're looking at. So this next table shows how long it would take for sterilization and at what temperature when we look at the difference between moist heat and dry heat. And you can see from this chapter, moist heat is much more efficient at a lower temperature and for a shorter amount of time. So that moisture plays a role at high heats that will actually sterilize much quicker. There are things to think about, though, when we look at heat, and there is some heat resistance, and that has to do often with spores, if you remember that, versus vegetative cells. Vegetative cells, again, are bacteria that are currently growing. So vegetative cells, even if they're spore formers, they're going to have the same susceptibility to vegetative cells of non-spore formers if they're in their vegetative state. So anytime any type of cell is in its vegetative state, it is much more susceptible to any of these controls. Pathogens are no different than non-pathogens in susceptibility to heat, and most fungi, protozoa, and worms are similar in their sensitivity to heat. The one microbe that we see that is pretty resistant to heat is viruses, and they're tolerant, you can see here, from 55 degrees Celsius, which is pretty warm, for 2 to 5 minutes, and even up to 60 degrees Celsius for 600 minutes. So viruses, it's probably not always the best way to do it, but it's something to keep in mind. Those vegetative bacteria cell would be very susceptible. So after heat, the other extreme of that would be cold, and often cold with desiccation or the drying out of the microbes. And this actually is not as good at killing the microbes, but it does slow the growth of microbes. And this is true in food during processing and storage. So once again, it just retards or slows down the activities of most microbes. And most of them are not long-term adversely affected by this cooling or long-term refrigeration or even deep freezing. And it's often a way that we actually use to preserve cultures of bacteria and viruses and fungi for long periods. So when they're in that cold environment, they're not growing at great speeds, but as soon as we then warm them from the cold temperatures, they usually are fine. So it's not one of the best ways to get rid of microbes. Another form that we use, the third form, is radiation. And radiation can be in a number of different types of rays or radiation. We have gamma rays, X-rays, and ultraviolet radiation. And these are often used with food products, things that won't hurt the food but will kill the microbe in it. So instead of doing heat that might ruin food or something else like that, radiation is sometimes used. After radiation, one that we have is filtration, and this is very effective both in air and liquids, and you can imagine that those are the 2 forms that we would have the easiest time filtrating. So with fluid, it can be strained through a filter, and it can have a filter that's large enough to let that fluid pass, but have too small of pores for those microbes. And what is often used is membranes of cellulose acetate, polycarbonate, and plastic. Those are often what is used. In addition, charcoal is sometimes used, and a couple of other different things can be used too. But pore size is what can be controlled to permit that sterilization or trapping of viruses or large proteins. So we have an ability to choose the size of pores for what we're trying to filter. And this is often used for things that can't take heat. Obviously, heat is a great way to sterilize things, but if we're looking at serum or blood products, any of these kind of medical things that we can't bring to high heats without either denaturing them or ruining their effectiveness, filtration is the way that we generally go. It's another way that we can decontaminate milk and beer. Often we do pasteurization, which is a high heat, but sometimes that can affect the taste of things. So this would be another option for that. And it's a very important step in water purification. So when we look at water purification, one of the major steps in it is filtration, of getting anything that they can that is larger than the pore size to be taken out. Unfortunately, it will not filter things that are soluble. So when you think about toxins that are released by bacteria, those could not be filtered out because they're too small. They'd get through the pores. So it's something to keep in mind when they're looking at filtration. And it's also a very efficient means of removing airborne contaminants. So you've probably all seen those HEPA filters that they use in hospital rooms. Some people even have them in their homes. And this just filters the air. It has a filtration system that the air flows through, and it traps things of a certain size that are bigger than their pore. So just the air gets through without the contaminants. And then the last one that we're going to talk about of these physical controls is osmotic pressure. And this is something that we've looked at in the lab already. In the lab, we've looked at adding large amounts of salt, but sugar's another one that can be used. And what it does is it creates that hypertonic environment. Remember, there's more solutes outside, so what ends up happening is the bacteria is going to try to have its water go out of the cell to try to dilute that solution it's in. And what it'll do is it will cause plasmolysis, which will make that bacterial cell shrink and often kill it. And this has been known for centuries, even before we understood bacteria. We've had pickling and smoking of meat and dry foods. Things like preserving of meat and different foods has been going on for ages because they've realized then that food won't spoil. And what causes the food to spoil? It's those microbes. However, osmotic pressure is never a sterilizing technique, so it doesn't go to the level of sterilization. So these are the physical controls that we see for microbes. My last lecture will be on the chemical controls. Hope this helps.